Imagine a life-saving medicine becoming contaminated because a tiny amount of residue from the previous product remained on the equipment. Even invisible contamination can lead to batch rejection, product recalls, regulatory observations, and risks to patient safety. That's why cleaning method validation is one of the most critical activities in pharmaceutical manufacturing. In this video, we will learn everything from the basic to advanced concepts used in GMP facilities. Introduction of CMV, a cleaning method validation. Cleaning method validation is the documented evidence that a cleaning procedure consistently removes product residues, cleaning agents, and microbial contamination to predefined acceptable limits. The goal is to ensure that equipment used for manufacturing one product does not contaminate the next product. Why cleaning validation is important? Cleaning validation protects patient by preventing cross-contamination. It also ensures compliance with GMP regulations, improve product quality, reduce batch failures, and build confidence during regulatory inspections. Sources of contamination. Residues may come from active pharmaceutical ingredients API, excipients, cleaning agents, lubricants, microorganisms, or environmental contamination. Even trace amount may affect product quality if not properly removed. Regulatory expectations. Global regulatory agencies expect scientifically justified cleaning procedures, risk assessment, documented protocols, validated analytical methods, and ongoing verification are key elements of a robust cleaning validation. Cleaning process design. A successful cleaning validation starts with a well-designed cleaning procedure. Every step should be standardized, including equipment disassembly, pre-rinsing, detergent preparation, washing, final rinsing, drying, and visual inspection. Critical parameters such as cleaning time, detergent concentration, water quality, temperature, and contact time must be controlled and documented. Consistency is essential because even a small variation can affect cleaning effectiveness. Swab sampling. Swab sampling is the most commonly used technique in cleaning validation. A trained analyst wipes a defined surface area using a validated swab motion with a suitable solvent. The swab is then extracted and analyzed, usually by HPLC, TOC, or another validated analytical method. Swab sampling is particularly useful for hard to clean locations such as corner joints, valves, and welds. Rinse sampling. Rinse sampling is performed by collecting the final rinse solution after cleaning. This method is especially useful for equipment with complex internal surfaces where swabbing is difficult. Although rinse sampling evaluates the entire system, it cannot identify the exact location of any remaining residue. Therefore, many facilities use both swab and rinse sampling together. Recovery study. Now, recovery study, it is most important parameter, recovery study. A recovery study demonstrates how efficiently the sampling method removes residue from the equipment surface. A known amount of the target compound, a known amount of the target concentration is applied to representative surfaces, allowed to dry, then recovered using the validated sampling procedure. The recovered amount is compared with the applied amount or added amount to calculate the recovery percentage. A consistent and scientifically justified recovery supports the reliability of the cleaning validation method. In the next part, we will cover analytical method validation. For cleaning validation, acceptance criteria such as MACO, visual cleanness, health-based exposure limits, validation protocols, report preparation, and the most common inspection observations from regulatory agencies.